Okay, welcome back. We are now on the 6th week of Molecular Endocrinology. We get back to the hypothalamic, pituitary, and the different axes. Now today we talk about IGF and growth control. And then the next 2 weeks we'll talk about the thyroid and also the reproduction. So here we focus on one thing, growth control. For growth control we have a very important hormone known as growth hormone. Okay, and growth hormone actually control IGF, insulin-like growth factor. So growth hormone, remember we have the different cell types in the pituitary gland, the acidophil and also the basophil. The basophil is the basophil here, including ACTH, luteinizing hormone, follicle stimulating hormone, or the gonadotropes, and then from the thyrotropes that produce thyroid stimulating hormone. And then the more acidic hormones are the prolactin and growth hormone. We talk about prolactin in the first week. And then we also talk about these different feedback loops or feedback mechanisms. Dopamine would inhibit prolactin and also TRH and MSH in the intermediate loop. And then hormone that would inhibit growth hormone is known as somatostatin. It's from the hypothalamus, but somatostatin also could be from the pancreas. The factors that will stimulate the formation of growth hormone, including these like TRH, thyrotropin releasing hormone, gonadotropin releasing hormone, GnRH. But the main hormone regulating the release of growth hormone is growth hormone releasing hormone, GHRH. So these three are from the hypothalamus. Now these are what we call those portal hormones because they are from the blood vessel system going down to the pituitary gland. Here in the acidophils there are two kinds of cells, lactotroops for prolactin, but somatotroops for growth hormone. Okay, and you are reminded that in the first week and the second week of my teaching, you may do your own homework with paper, putting down what are the factors from the hypothalamic gland, what are the hormones from the pituitary gland, and then what they do or where they go into the organs. In this case, growth hormone would affect the organ known as in the liver, hepatocytes. And then there's several different effects, and that would make yourself a clearer picture by drawing your own chart. So we actually have to divide this into 4 videos, perhaps video 1 and 2 on the background and also growth hormone and its receptors and action. And then we have video 3 focusing on IGFs, and then we have the last video on mainly growth control. So let's start with growth hormone. Growth hormone is a very big hormone. It has a complicated structure with these A to B and then C and then flip around here and D. So they have a very complicated what we call anti-parallel folding of 4 alpha helical bundle fixed with 2 disulfide bonds. The genes are all encoding for these peptides in one polypeptide with A, B, C, and then D. And then if you do this plot, you will find these are more hydrophobic, so they form hydrophobic loops at A and B and C and D. And then in between the alpha helical structure, there are loops that will link them together, and then you can see this is from the N terminal. This is A going down here, and then flip over to B, and then B up to C, and then C turn down into here, and D. So therefore there's A, B, C, and then D. We call it anti-parallel folding. And this is a very complicated structure that took scientists almost 2 decades to resolve its structure. And if you take a closer look at this diagram taken from this old paper by Cunningham and Wells, 1989, you can see this antiparallel structure with A, and then fit into B and C, and then fit into here, D. The last alpha helix going down into, this is the tail, with a short disulfide loop here forming this structure like this. Another disulfide loop is in here. The dots are those mutations that they have done to confirm the structure by changing all these different amino acids into alanine. You know, alanine has no side chain. Side chain is only CH3. Therefore, by removing the chemical properties of the side chains of these amino acids, they determined that these are important sites for the growth hormone to bind to its receptors. And surprisingly, they found two sites, site 1 and site 2. So meaning that the two sites are required to bind to the growth hormone receptor. And more exciting data coming out a few years later, that the growth hormone would form this structure with the anti-parallel alpha helical domains A, B, C, and then D. And then with that, they actually with site 1 and 2 to bind to two different receptors. Well, I should say two receptors. They're the same receptors, but then they are actually GHR1 and GHR2. Or one growth hormone binds to two receptors. Another interesting thing is that we also found that in the blood or in the plasma, they have these what we call binding protein. We call them growth hormone serum binding protein. The binding protein is exactly the same as the extracellular domain of the receptor. In other words, the growth hormone, when they are in the bloodstream, they are bound with the serum binding protein. Okay, and then when they go into the receptor or into the target sites, they would have to release the growth hormone and put it onto the growth hormone receptor. Okay, so the extracellular domain of the growth hormone receptor is the same as the growth hormone serum binding protein. That's what that means. And then when they bind to the growth hormone receptor, so these are the two sites, and site 1 and site 2 I've just mentioned. So the serum binding protein would take this growth hormone with the binding sites 1 and 2, and then to bind to the receptor. You can see the receptor is here, maybe I use this here. And then the membrane growth hormone receptor, and then this binding protein would go away, and then the growth hormone would bind to here, and then it requires dimerization. So at low concentration of growth hormone, successful dimerization, the receptor would be activated. But if you have too high the concentration of growth hormone, you would dilute out the receptors. Say like this is an example that about activity here, and then growth hormone concentration, increasing growth hormone concentration. It actually has what we call an optimal level of growth hormone. The optimal levels is here. Higher concentration, the activity will drop. So it forms this what we call bell-shaped dose response curve. Okay. So why do we need growth hormone binding protein in serum? I would say that this is for the protection of the growth hormone because it has a very complicated structure with disulfide loop and so on, and also to avoid, degradation. The second is that it would bind like this to the receptor, so it would present the hormone into its receptor. Okay, but still there are more mystery than what we have known. So how would this activation of growth hormone receptor sustain? The answer is not really sustaining. It requires dimerization. After dimerization, the kinase inside there is called JAK kinase or Janus kinase. What is Janus? Janus is that the protein with two phases. It would bind together, and then it would turn into autophosphorylation of the receptor itself. Autophosphorylation means that the kinase of Janus would phosphorylate its own protein, so that is the receptor. At the same time, this JAK kinase would phosphorylate its target protein, known as ST-AT. And then this ST-AT would add on, going into nuclear translocation to add on the target genes inside the nucleus. We'll come back to that slide later. So in other words, you may also think of, so what's next? Why do they need to autophosphorylate themselves? Okay, number one is for signal transduction. Number two, what happened after dimerization? Could you think about this? I'll come back to that later. Now this is the reactions coming in. JAK2 phosphorylation is going on to stimulate MAP kinase, RAS protein. These are other growth factor signaling pathway. And then the ST-AT I've just mentioned is known as signal transducer and activators of transcriptions. There are ST-AT 1, 2, 3, 4, 5, many of them. To make this slide more easier to read, we have ST-AT, and ST-AT will go to transcribe other genes to be expressed. These genes including one of these key growth-related oncogene called C-force. And also AP1 is an activating promoter site with GLE2 or 1 location relating to ST-AT1 and 3 and 5 here. And then we have this steroid factor and so on. So a complicated picture putting together in simple terms is that growth hormone will stimulate JAK2. This is a Janus kinase with phosphorylate themselves, and then it's phosphorylate ST-AT. ST-AT also stimulate other genes to express for growth control, of course. And then this pathway also stimulate this PI3K, bring about glucose import, and also on the membrane would stimulate proteins kinase C pathway. And these also will bring in calcium signaling to stimulate more gene transcription. How to stop this pathway? By dimerization after phosphorylation, the growth hormone would be engulfed inside the cell by endocytosis. Okay, so therefore that will be stopped, and then those stimulated factor will stimulate a lot of gene transcription. So dimerization, phosphorylation after that, dimerization, and then growth hormone receptor is here. There's a growth hormone 1, growth hormone 1 to 2 receptors, and then JAK2 will stimulate this step 1, 3, and 5. Actually, the step 1, 3, 5 will also form hormone dimerization, go into the target genes. Target genes would induce, for example, in the liver and fibrosis, will stimulate IGF-1, insulin-like growth factor 1, and also protein synthesis, lipolysis in adipose tissue, amino acid transport in liver, muscle, and adipose tissues, and also carbohydrate metabolism or hypercalcemia effect in the liver. Okay. Well, in the final examination, you are going to challenge to provide many different short MCQs. Like this is one of them. Show you occasionally. Which one of the following is a correct description of human growth hormone? Okay, pay attention. A, GH is a protein hormone from the neural hypophysis. Well, I mean, it's one correct, right? Obviously, this one is incorrect because growth hormone is not in the posterior pituitary gland, it's in the anterior pituitary gland and also in the acetophil. So it's not from the neuron, it's from the portal hormone, portal hormone. Growth hormone activates IGFs in the brain. Growth hormone binds to a dimer of a specific membrane receptor on target cell to excite its function. Circulating GH has its lowest level at night. Growth hormone secretion is inhibited by growing. If you have to select, there will be one correct answer, right? So as mentioned before, growth hormone binds to a dimer of specific membrane receptor. So you just ignore the other 4 because this is the one that you have studied before, right? So growth hormone binds to the dimer of a specific membrane receptor. One growth hormone binds to 2 receptors. Okay. Growth factors is correct, but not in the brain. The brain cannot be regrown. So if you find IGF in the brain, that could be glioma or brain tumor. Same thing is true that growth hormone lowest level at night is also not correct. Growth hormone is always very high at night. We probably need to go to sleep and then we have our body growth and we sleep. Okay, growth hormone secretion is actually stimulated by this growth hormone secreting stimulators called growth. We'll come back to these different things later. Growth hormone, when they come out, as I mentioned, that the way that we remove the growth hormone is by endocytosis, engulfing the growth hormone in. So the growth hormone actually are being produced and then secreted from the pituitary. Interestingly, in what we call a pulsatile form. Pulsatile form means pulses. Oh, this is pulses. Pulses. So the time over from 0 to 1500 minutes, at least for this study, pulsatile hormone secretion in normal human. You withdraw the blood and then it would form these pulses, right? So in other words, when you detect a growth hormone, you cannot just detect at one time point. You may need to collect blood from different time points. Another feature that is of importance is that you can see that young people, growth hormone level, okay, would be pulsatile, that probably at night, and then more growth hormone come out. But then for the elderly, old people, you do not see that pulsatile form. Well, some pulses, but in general, the level is low. What that is to say, young people have higher growth hormone level because those young people, they need to have somatic growth. But for the old people, old folks, elderly, you do not need to have somatic growth anymore. So the growth hormone level actually dropped. At one point, around 30 years ago, 40 years ago, people even promote the use of growth hormone to the elderly so that could rejuvenate their lifestyle. I mean, some patients would ask for that kind of help. But obviously, this is beyond molecular endocrinology. But what I'm trying to explain to you is that because growth hormone also induces IGFs, so if there are too much growth or somatic growth or too much growth factors, it would also cause tumors. So it may not be a good thing to use growth hormone. Now, growth hormone also came out at night. This is a chart that showing the diurnal changes of hormone in young adults. Cortisol level may be high when you wake up in the morning. Aldosterone fluctuating, testosterone quite normal. Luteinizing hormone or follicle stimulating hormone, not change much. And thyrotropin also come a little bit at night. Prolactin in the morning, but they slow down. But significantly, you can see the curves here. Growth hormone in pulsatile form, but then at night they come out significant. That is to say, perhaps we need to pay attention to the growth hormone at night. For young adults, that the somatic growth is actually relating to a very good sleep. Sometimes we go to sleep, and very good sleep at night, and then we need to have growth hormone to facilitate growth at that time. And then we also think about the mechanism of pulsatile nature of growth hormone secretion. It's actually not very well understood. We have the control here, and then we also compare fasting. You can see the fasting. So if you eat too much, it may not be a good thing that to promote growth hormone production. You can see the fasting, especially for young people. fasting will stimulate growth. Okay, so eating too much may not be a good thing. So this is to try comparing 24-hour growth hormone production rates, not because they are impulsive type, right? So they accumulate the levels of those growth hormone production in 24 hours, observed in 6 different individuals, and 9 young men on a fat day, and also second day of a fasting, only fasting for 1 day. You can see a significant increase of growth hormone production. And then a similar study also compare older men fat, older men fast. You can see more growth hormone stimulations, regular growth hormone secretion rate, not fasting. You can see here another patient also fasting, and then more growth hormone. And then for women, same thing is true. And well, occasionally some spikes there. This for the fat one, and then for the fast, you can see more growth hormone stimulated. Okay, so perhaps we should understand more on the secretion of growth hormone, the role of growth hormone in the elderly. Okay, and then there are also other questions posed, like for example, the animals being treated by genetic engineering or recombinant growth hormone protein. And usually those farm animals will produce lean meat. In other words, growth hormone will promote lipolysis. Okay? And then we come back to that question, should the elderly be treated with growth hormone? The concern is that that would stimulate growth factors, so that may not be a good thing. So let's take a look of more stories on growth hormone. This time is with foods, appetite, and also hormones. There are also other hormones we mentioned last week on leptin. We also have ghrelin. These are what we call appetite stimulator. They also suppress. Ghrelin is actually the factor counteracting the effects of leptin. Leptin is oxygenic effects. Okay. So it's from the adipose tissue, so leptin is from the adipocytes. As appetite suppressors, because if you're fed up with fat and then secreted by the fat tissue, you tell the brain, okay, we have enough food, so don't eat more. Another one is oxygenic, is ghrelin. Ghrelin is appetite stimulator, and then when you are hungry from the stomach, and then from the stomach, that will send this ghrelin to your brain. And interestingly, ghrelin also induces growth hormone. And same thing is true that leptin is also stimulating growth hormone. So what does growth hormone do? And they have the receptors in the hypothalamus. I mean, the ghrelin, this now is known as growth hormone secreticons. There is a receptor there. And then in the hypothalamus, also from the hypothalamus, not just from the stomach, but also from other parts of the brain. The same thing is true that leptin, that would stimulate, and then for different appetite control, but they have counteracting effects of body weight and also appetite effects. Okay, and so more and more of the recent research are now on this area, trying to understand how these two hormones control our appetite and also control body weight. And then we come into more details of the two peptides. GHS is known as growth hormone secreticons. And we, on the other hand, don't forget that we still have the GHRH growth hormone releasing hormone acting from the hypothalamus on the pituitary gland to stimulate cyclic AMP and the release of growth hormone. And at the same time, we have synthetic GHS, or the one from our stomach or from the brain is called the ghrelin, and that will stimulate GHS out. This ghrelin has these amino acids with a special side chain here, So there are many synthetic GHS is being used to stimulate calcium signaling and stimulate the release of growth hormone. And so these are also very important drugs for the treatment to induce growth hormone, not necessarily prescribing using injections of growth hormone like 20 or 30 years ago when it first came out as synthetic or recombinant growth hormone produced by a very famous biotech company called Genentech, and that growth hormone would stimulate growth, and then now they actually we replace growth hormone by using GHS or synthetic growth hormone secreticons. Okay. So the effects of growth hormone on growth and metabolism, we have a conclusion that it would promote bone elongation. Okay, so growth, what's the meaning of growth? There are different meanings of growth. One we consider height is also an important factor. So for the height, so bone elongation, like for example the thigh, the legs, or the limbs. Okay. Second, protein synthesis and tissue growth, and also including erythropoiesis. Erythropoiesis means production of red blood cells or red cells. Okay. And then number 3, decreased blood amino acid concentration and decreased urinary urea nitrogen. And actually the nitrogen or amino acids are being used for protein production. 4, GI absorption of calcium. So the gastrointestinal tract will need more calcium for bone elongation, right? Decreased cellular uptake of glucose, increased hepatic output of glucose, leading to hyperglycemia because we need to have more metabolism. So we need to have more sugar. Hormone lipolysis in adipose tissues because we want the adipose tissues, we want the lipid to be converted back into sugar. And that we need this energy for growth and metabolism being activated. Okay, so with that we stop, and then when we come back in the next video we talk about IGFs.